So good morning. So today I am going to discuss about structure of antibody. So immunoglobulin or glycoprotein formed in response to an antigen and react specifically with that antigen. They are commonly called antibodies. They are found in serum, body fluid and tissues. They are synthesized by B lymphocytes and secreted by plasma cells. Randy Porter in the year of 1962 proposed the basic structure of antibody. It is Y-shaped in nature. It is made up of four polypeptide chains. There are two types of chains. One is light chain, another one is heavy chain. Antibody has a two light chains. They are shorter and identical. It is made up of 214 amino acids. Based on the structure of amino acid sequence, it is grouped into two types, namely kappa and lambda. Approximately 60% of serum immunoglobulin molecules are K-type, while only 40% are lambda type. Heavy chain. It has two heavy chains. They are longer and identical. Each heavy chain is made up of 450 to 700 amino acids. Heavy chains are five types. They are gamma chain, alpha chain, mu chain, delta chain, and epsilon. The four chains are interconnected by disulfide bonds. Each light chain is linked to a heavy chain by a disulfide bond and by non-covalent interactions such as salt bridges, hydrogen bond, and hydrophobic interactions. One end of each chain is called amino terminal end or N-terminal end, and another end is called carboxyl terminal or C-terminal end. Variable and constant region in antibodies. Light and heavy chains are subdivided into variable and constant region. Each heavy and light chains contains amino terminal in variable region, carboxyl terminal in constant region. Variable region extends from N terminal about 110 amino acids, and amino acid sequence in this region is highly variable. It is an antigen binding region. Constant region. It is extends from end of variable region to C terminal, and amino terminal sequence is relatively constant. CDR region. Each variable domain of the immunoglobulin at its tips contain three variable loops for binding the antigens. These loops have complementary sequence of amino acid with that of antigen. These regions are called complementary determining region or CDR region. It is also referred to as hypervariable region. Domains. Light chain has two domains. One is variable light chain domain. Another one is called constant light chain domain. Heavy chain has four domains. Variable heavy chain domain, constant heavy chain domain that is called CH1, CH2, CH3. In immunoglobulin M and immunoglobulin E have additional constant heavy chain domains called as CH4 domain. Hinge region. The heavy chain has a central flexible region connected by disulfide bond or called hinge region. It consists of about 12 amino acids located between CH1 and CH2 domains. It contains many hydrophilic and proline residues. Hydrophilic residues makes peptide bond in unfold. Proline residues make the chain flexible. Pepsine and pepine act in this region. Immunoglobulin E has no hinge region. Fab and FC region. When immunoglobulin is digested with proteolytic enzyme called pepine, It splits at hinge into two fragments, namely Fab region and FC region. Fab region is called antigen binding fragment. FC region called crystalline fragment. Papain cleavage occurs between the disulfide bond of hinge region. It produces three fragments, two identical fragments called Fab fragments or antigen binding activity. Other fragment is called FC fragment or fraction crystalline. Thank you.